Okay, so the title of today's research presentation, Sickle Cell Disease Using Deep Learning. This is Professor Dr. Ahmed Wasif Reza from Department of Computer Science and Engineering, East West University, Bangladesh. So this is my email address. So this is my personal detail. And here I mention my department and also my email address, my homepage, then Google Scholar, Web of Science, Scopus, ORCID, PubMed, ResearchGate, and my office address is mentioned here. So this is my short biography. I've been working in the fields of wireless communication, biomedical image processing, bioinformatics, artificial intelligence, machine learning, deep learning, robotics and Internet of Things, green computing, IT, and sustainable development and biomedical image processing for many years, both in industrial exposure and academically research valued work. I have authored and co-authored several science citation index, Web of Science, and Scopus Index journal and conference paper, more than 220 papers so far. I received the Best Researcher Award, Outstanding Championship Award, Excellence Contribution Award, Best Paper Award from various international conference and organization for my outstanding contribution to research and publication. According to the AD Scientific Index 2024, I am recognized as computer science best scientist in my university and top 3% scientist in computer science among the scientists in the AD Scientific Index in the world or country. Now, what is sickle cell disease? Sickle cell is a genetic disorder brought on by a beta globin gene mutation leading to abnormal hemoglobin formation. As it is seen from the samples, hemoglobin is the main component of red blood cell. Blood disorders such as sickle cell anemia and diamond blackfan anemia are genetic disorders that may have an impact on the body's ability to produce red blood cells. So what can happen? It can shorten lifespan, impair many organ systems, affect the kidney, and result in permanent disabilities. So it may also result in severe complication, including damage to the eyes, heart, lungs, liver, kidneys, and joints, increase susceptibility to infections, and acute chest syndrome. Even for children, sickle cell disease causes acute chest syndrome and gallbladder disease. So my other biomedical researches ongoing, which is on that is heart, lungs, liver, kidneys, and so on. So this is blood cell samples. It is seen from the slide. You can see that is circular, elongated, and others. Three categories of individual cell image are shown. So first of all, circular image, that is called red blood cell RBC samples. The next is elongated image, that is called deformed RBC, that is the sickle cell, and other images, that is sample of images other than RBC and sickle. So sample images in the data set, what we collected actually, this is shown in the slide. You can see source image, label image, mask circular, mask elongated, and mask other. So this is the methodology. If you see from the figure, this is the overview of the proposed methodology. So first of all, image acquisition, then individual cell separation, then image augmentation, then image pre-processing. Then we can see hyperparameter tuning and ablation experiment. After this, we can see machine learning classifiers are involved, and then classification. Classification of what? Classification of circular, elongated, and others. So this is the overview of the proposed methodology. So this is deep learning architectures and machine learning models. So what are the models? We use ResNet 50, AlexNet, VGG16, VGG19, and MobileNet. And also for the classifier, we use random forest classifier and logistic regression. So here in this slide, you can see ResNet 50 architecture, then AlexNet architecture, VGG architecture, and MobileNet architecture. All this architecture are newly proposed by tuning new parameters. So these are all our proposed architectures for the proposed sickle cell disease classification techniques. So now, performance of hyperparameter tuned models. You can see from the figure, it is seen clearly that ResNet 50 achieved a test accuracy of 97.71% and a validation accuracy of 96.95%. Then we can see VGG16 achieved a test accuracy of 98.96% and a validation accuracy of 98.27%. Then VGG19 achieved a test accuracy of 99% and a validation accuracy of 98.58%. MobileNet achieved a test accuracy of 96.99% and a validation accuracy of 94%. Then AlexNet, according to the batch size, we can see batch size 32. AlexNet achieved a test accuracy of 34.36% and a validation accuracy of 34%. Then AlexNet for the batch size 3, we achieved a test accuracy of 99.13% and a validation accuracy of 94.34%. So this is the performance of the hyperparameter tuned models. Now come to the design of the ablation experiment. This is also a tremendous contribution of our research. We can find the best performance from the MobileNet and SVM, that is support vector machine. We found validation accuracy of 99.53% and test accuracy 98.90%. Then second best, we found ResNet 50 and SVM. We found validation accuracy of 99.32% and test accuracy of 98.79%. Then third best, we found VGG16 and SVM, that is validation accuracy of 98.71% and test accuracy of 97.84%. So we see from this ablation experiment, that is a transfer learning architecture is shown here. Input, we can see the input, then the transfer learning architectures, then transfer learning hyperparameter tuning, then batch size, learning rate, activation function, dropout epoch, all are shown here. For the detail, you have to see my publication. And then machine learning classifiers, that is we use random forest and support vector machine, then machine learning hypertune, and then output. So this is the ablation experiment design. So finally, to conclude my research, we can see that this research actually presents a transfer and deep learning based approach to the classification of sickle cell disease. 5 transfer learning models such as ResNet 50, AlexNet, MobileNet, VGG16 and VGG19, and a sequential convolution neural network, that is CNN, have been implemented for sickle cell disease classification. An ablation experiment using random forest and support vector machine classifier along with various hyperparameter tuning was carried out to determine the contribution of different model elements on their predicted accuracy. Additionally, a rigorous statistical analysis was carried out for evaluation and to further evaluate the model's robustness. An adversarial attack test was conducted also. The experimental result demonstrate compelling performance across all models. So after performing the statistical test, it was observed that MobileNet showed a significant improvement, while other models, for example, ResNet 50, AlexNet, VGG16, and VGG19 did not. So P is shown here in this slide. Notably, the ResNet 50 model achieves remarkable precision, recall, and F1 score values of 100%. This is tremendous achievement, 100% for circular, elongated, and other cell shapes when experimented with a smaller dataset. The AlexNet model achieves a balanced precision, that is 98%, and recall 99% for circular, elongated shapes, So meanwhile, the other models showcase competitive performance. So we'd like to thank this UGVIA research group. The dataset is collected from this UGVIA research group from Spain. And this research work is published as follows. It is shown in the slide. It is published in Helion, okay, Elsevier and Cell Press, which is indexed in Science Citation Index, Web of Science, and Scopus Index. So my other research on biomedical, I want to also present. That is tuberculosis bacteria detection using deep learning, which is published in lecture notes in Network and System recently. All these are published recently that I want to show here. And next one is innovation method for Alzheimer's disease detection using convolution neural network. This is also published in Springer Nature in lecture notes in Networks and System. This is also my recent publication. Another publication, MRI based brain tumor classification using various deep learning convolution network and CNN. This is also published in Springer Nature, lecture notes in Network and System. Another publication, a computer assisted detection framework of kidney disease based on CNN model. This is also published in Springer Nature, lecture notes in Network and System. This is another publication, a CNN-based strategy to classify MRI-based brain tumor using deep convolution network. This is published in Applied Sciences, MDPI. So this is another publication, recent publication, that is combating COVID-19 using machine learning and deep learning, application, challenges, and future perspective. So this is published in Array Elsevier. So my research profile has been selected for the International Research Awards on New Science Invention, that is called NISIN Awards, under the category of Best Researcher Award for my remarkable contribution to digital health and biomedical engineering research. So I sincerely thank Science Father and the NISIN Awards organizing committee for selecting me for this prestigious award and recognition. Thank you, everyone.